Hello everyone and welcome to the Precision Medicine virtual event. My name is Raed Samara. I'm an Associate Director in the Global Product Management team at QIAGEN, and I will be presenting on the QIASeek Expanded Carrier Screening Panel. Please feel free to submit questions during my presentation and I will follow up with you via email. This presentation will be broken into two pieces. I will go through the chemistry part of the panel, and then Rupert will jump in to speak about the bioinformatics piece. And to start off for the chemistry part, I thought we would start off with the issues that we commonly see in library preparation approaches. I've broken down the issues into four main categories: primer dimers, specificity of enrichment and sequencing, the uniformity of enrichment and sequencing, and PCR duplicates and errors. And then next, I basically explained why these issues are significant and what the consequences are. Basically, for primer dimers, what you end up with is a lack of coverage across your regions of interest. Why is specificity of enrichment an issue? It's because it would be associated with a higher cost per sample. And the same goes for uniformity. If you have a low uniformity across your regions of interest, you end up with a higher cost per sample. PCR duplicates and errors are a problem or an issue simply because they could end up with inaccurate variant analysis. So what we've done at QIAGEN to overcome these issues and consequences is to build some solutions that would address each one of these issues. And these solutions are part of what we call the QIASeek Solution. And these solutions are basically a single primer extension approach for enrichment, which can get over the primer dimers issue. And I will go into single primer extension and all of these elements and solutions in subsequent slides. And basically, to overcome the issue of specificity and a higher cost per sample, we've built industry-leading primer designs that allow us to achieve a very high degree of specificity across the regions of interest. And in order to also overcome the issue of uniformity, we're incorporating single primer extension approaches for enrichment that will allow us to achieve a higher degree of uniformity, as you will see in subsequent slides as well. And to overcome the issue of PCR duplicates or errors, we've incorporated unique molecular indices, or UMIs, into our library structures. With UMIs, we can now go back to, with powerful data analysis tools, and analyze data not just basically based on raw reads, but we can now analyze data based on unique molecular indices, which will allow us, again, to achieve a much more accurate variant analysis, as you will see in subsequent slides. To highlight how single primer extension approaches work, I thought I would compare a single primer extension approach, which is a spin on amplicon type of approaches. I would compare how single primer extension approaches work compared to the traditional two-primer amplicon design. And so on the left-hand side, you see the two-primer amplicon design, in which two region-specific primers are required to enrich for that specific region. So for instance, if you're interested in exon 21 of EGFR or any other gene for that matter, with a two-primer amplicon design, you would require two EGFR exon 21 specific primers, one that flanks the beginning of the site that you're interested in, and one that flanks the end of that specific region. Now, the outcomes of such an approach, while this has been a widely used approach, there are several limitations of such an approach. First, you end up with an increased risk of primer dimers and dropouts, and what this translates to is, of course, you end up not covering that specific region, or you end up covering it very poorly. Another issue that we see is a lower degree of uniformity. In a lot of instances, if you have continuous stretches of genomic regions that you want to cover, you end up with multiple pools of primers that would be required to cover all of the regions that you're interested in. And you end up with a low library complexity simply because your start and stop positions are defined by primers. And now increasing the panel content might require primer redesign. Again, this goes back to that very first point of increased risk of primer dimers. If you want to increase the panel content, you would have to come back and basically ask the question, for those new primers that I'm adding into the panel, how do they affect the performance of existing primers? So the solution to this kind of issue is the single primer extension approach that we've incorporated into the QIASeek Solution, or SPE. With single primer extension, basically, it still is an amplicon-based approach. However, when you are trying to define your regions of interest, you don't need two primers to define a region of interest anymore. All you need is just one region-specific primer. The other primer that will be used for the amplicon-based approach is basically a universal primer that binds to sequences introduced in the library adapters. And so if I go back to exon 21 of EGFR as an example again, I do not need to define exon 21 of EGFR with 2 primers anymore. All I need is just one EGFR exon 21 specific primer, and then the other primer will be the universal primer that will bind to the library sequences, or the sequences introduced in the adapters. What this allows us to do now is to lower the risk of primer dimers. It allows us to increase the uniformity of enrichment and sequencing, again, as you'll see in a little bit. And this also allows us to pool all of the required primers into a single pool of primers. So you do not need to split your primers into multiple pools anymore. It allows us to achieve a higher library complexity because it's only the start position that is defined. The end position of enrichment is going to be defined randomly by this universal primer. And now increasing the panel content does not require a complete redesign of existing primers. You can, if you decide to, for whatever reason, if you decide to increase the content of your panel, you don't have to redesign existing panels anymore. You can just take the new primers that are required for the new genes or new regions of interest and simply spike them into the existing primers. So that way, increasing the panel content does not really require a complete rework of the existing primers. So how do unique molecular indices work first? So I've gone over how single primer extension approaches differ from the traditional 2-primer enrichment approaches, and what I would like to do next is to go over how unique molecular indices allow us to achieve a higher level of quantification of variants and transcripts, as well as how it would help us get over the issue of duplicates, PCR duplicates, and errors. So when it comes to quantification, unique molecular indices can help us achieve this higher level and more accurate type of quantification, because we are not counting just raw reads. We are actually counting unique original molecules that have been tagged by unique molecular indices before any enrichment takes place. So if I use this example of trying to quantify, let's say, transcripts mapping back to, or reads mapping back to exon 21 of EGFR, in order to get an accurate quantification level of the EGFR gene. On the top part of this slide, you see the scenario where we're using just simple raw reads as a way to count the levels of EGFR based on exon 21. The issue that we see with this type of approach is that when you look at these 5 reads that map back to exon 21 of EGFR, you really cannot tell whether these represent 5 unique DNA or RNA molecules, or they're just basically quintuplets of the same DNA or RNA molecule that was just amplified over and over. So you can't really tell whether you're looking at PCR duplicates or true fragments. With unique molecular indices that you introduce before any amplification takes place, and I'll go through the workflow in a little bit, when you introduce unique molecular indices before any amplification takes place, what you're doing is you're tagging the original molecules with these unique molecular indices. And when you look at your data with bioinformatics, sophisticated bioinformatics tools, you can actually analyze the data not based on the raw reads anymore, but also on unique molecular indices. And the way this works is basically, if you see, as an example, if you see 5 unique molecular indices as indicated in colored fragments, if you see on the left-hand side, if you see 5 unique molecular indices across all of these reads that map back to exon 21 of EGFR, you've started off with 5 unique DNA or RNA molecules. So these are not PCR duplicates. They represent 5 unique molecules. However, if you end up with 5 reads that carry the same unique molecular index, you've started off with just 1 transcript, basically, that you've amplified over and over, and you've not started with 5 unique molecules. You actually only started with 1 that was duplicated over and over. So by incorporating unique molecular indices before any amplification takes place, you now have a way to differentiate between what is a true molecule, a true DNA or RNA molecule, and a PCR duplicate. Now, how do unique molecular indices allow us to achieve a higher level of variant calling? It's pretty much the same sort of principle or procedure. You take your unique DNA molecules and you assign or ligate unique molecular indices onto the unique fragments, basically, before any amplification takes place, in order to take advantage of unique molecular indices. So if I use, again, the same example of exon 21 of EGFR, and I'm trying to, let's say, quantify variants that map back to exon 21 of EGFR, on the top part of the slide, you see an example of how you're using raw reads to try to quantify the variants that map back to exon 21 of EGFR. And again, if you see one mutation or one variant popping into an individual read out of 5, with raw reads, it's very difficult to discriminate between this as being a true, let's say, low-frequency mutation, or whether it is a PCR error that was introduced along the workflow. So in order to overcome this issue, we use, again, unique molecular indices that are introduced into the unique fragments before any amplification takes place. And what UMIs allow us to do now is to actually differentiate between a true low-frequency mutation or an error that was introduced along the workflow. So on the left-hand side, you see an example of a false variant or an error that was introduced in the PCR steps. And the reason being is because all these 5 fragments carry the same unique molecular index, but we only see the mutation in, let's say, 1 fragment out of 5. If this was a true low-frequency mutation, you should see the variants in all of those fragments that carry the same unique molecular index as you see on the right-hand side. So by incorporating unique molecular indices, we now have an approach to discriminate between what is a true low-frequency mutation and what is a PCR error. And now to show you the advantages of the single primer extension approach, I mentioned that you can achieve a higher level of uniformity and coverage across different regions. Just to give you a couple of examples, I'm showing you here some data that compares a single primer extension approach versus a 2-primer amplicon approach for enrichment. And I'm using NRAS as an example. We're looking at enrichment of exon 3 of NRAS here. And what you see on the top part of this graph is the type of coverage that you would get using a 2-primer amplicon approach versus single primer extension approach which has been introduced in the QIAGEN workflow on the bottom part of the slide. And then in the table, you see the numbers represent basically the read depth across a specific region that we're looking at, and you're also looking at the coverage range. So if you look at how exon 3 of NRAS has been covered using the 2-primer amplicon approach versus the single primer extension approach in QIAGEN, you do see big differences. First, the 2-primer amplicon approach, because of the issue of primer dropouts and primer interactions, you will see a big difference in terms of the read depth across the different regions of exon 3. So for instance, half of exon 3 of NRAS has been covered at 255x versus the other half being covered at 12,000x. So it's a big difference when you look at the range of coverage. However, if you look at how exon 3 of NRAS has been covered with the single primer extension approach in QIAGEN panels, you do see a much tighter range, and you see that all of the bases have been covered fairly uniformly. So the lowest type of coverage that we see for exon 3 of NRAS, for example, is 872, but then the maximum type of coverage is 2033x. So again, what this allows us to do is to achieve a higher degree of uniformity across the regions that we want to enrich, and this actually now could drop the price per sample because you don't have to account for the lowly expressed or lowly covered regions that you see, let's say, in a 2-primer amplicon approach. And then this also allows us, the single primer extension approach allows us to achieve a higher degree of uniformity even compared to, let's say, a hybrid capture approach. So if you compare the type of uniformity that you see at different depths of coverage, as you see on the right-hand side of this graph, these are different levels of coverage all the way from 1x to 160x. And on the x-axis, you see different types of genes that have been interrogated, and on the left-hand side, you see basically the percent of bases of these genes that have been covered at a specific depth of coverage. So if I look at the very first row at 1x, you do see fairly uniform coverage, whether you're using single primer extension approaches or hybrid capture approaches. And the same is true for 10x coverage, 20x coverage. But as you increase the coverage across your regions of interest, you start seeing a lower degree of uniformity in hybrid capture approaches compared to the single primer extension approach in the QIAGEN panels, which basically tells us that the single primer extension approach in the QIAGEN panels is going to allow you to basically achieve a higher degree of uniformity, which means that your genes are going to be more uniformly covered regardless of the type of genes that you go after. So how does the workflow look like? I mentioned before that we assign unique molecular indices before any amplification takes place, and this is shown here at a high level. Basically, you take your input DNA, and the workflow tolerates as little as 10 nanograms of input DNA. That DNA gets fragmented enzymatically in a single enzymatic reaction to basically produce smaller fragments. The smaller fragments are then also end-repaired and A-tailed before library construction. The next step after fragmentation is unique molecular indices assignment and library construction. This is where you are ligating adapters to the ends of the DNA fragments that you generated in the first step, and these adapters also contain unique molecular indices. So in this second step, you're basically constructing your library to include unique molecular indices. And then in the third step is the single primer extension approach for enrichment. So you've generated your libraries, and then in the third step, you're going to enrich for your regions using a single primer extension approach. And remember, all of the primers that are required have been pooled into an individual pool of primers. And once you've enriched for your regions, the last step is to go through library amplification, where you can multiplex up to 384 samples per sequencing run. And in the entire workflow, going from extracted DNA all the way to libraries that can be quantified and sequenced, is basically a one-day workflow. And last but not least, the way we've built these solutions is basically a sample to insight approach, which means that we're not only providing the panel itself and the indexes that are required for library construction, but the chemistry is always married to analysis pipeline, sophisticated analysis pipeline that can analyze data not just based on raw reads, but also unique molecular indices, as well as interpretation tools. So if you look at, let's say, the expanded carrier screening panel, this sample to insight approach that we've taken for this type of panel will basically give you access to proprietary PCR chemistries to enrich GC-rich regions in the panel box. And you also have primers that are based on single primer extension approaches for enhanced uniformity. In addition to the panel box, you also need an index box. This is the sequencer-specific part of this workflow. The panel box itself is actually sequencer agnostic in the sense that you could use the same chemistry for enrichment, whether you're using the Illumina platforms or Ion Torrent platforms. However, the index box is sequencer specific in the sense that you have to choose an index box that is compatible with your sequencer. And this box contains the molecularly barcoded library adapters to incorporate unique molecular indices. And as you'll hear from Rupert, the analysis pipelines, they do analyze data based on unique molecular indices, and there are sophisticated interpretation solutions to allow you to take the data that you generated from the panel and interpret it for your purposes. I will turn it over to Rupert now.
Thanks, Raed. As my colleague Raed has so eloquently described, the upstream sample prep is a very optimal design to generate high-quality raw reads and raw data to be interpreted by our informatics systems. So when we talk about bioinformatics, people think of a lot of different things for bioinformatics, but today what I want to talk really about is about bringing knowledge to help interpret the information that is gleaned from the sequencing of DNA samples. If we take a look at the universe of possible human mutations, we're just now embarking on the genomics revolution, and so our knowledge is constantly growing. Now, no one person can obviously know everything there is to know about the entire domain of human genetics, and so your own knowledge may be limited to a specialized domain. If you use public crowdsourced databases such as ClinVar, you get a lot more information, but yet this is not representative of all there is that is known to mankind. There are other databases such as HGMD, which then attempt to curate the entire corpus of scientific literature to identify disease-causing variants. And what I want to show you is that QIAGEN has a proprietary knowledge base that encompasses not just public information, but proprietary information, as well as its own efforts to try and bring you the most complete picture of known pathogenic variants that is available to mankind. Now, how do we do this? Well, we've been doing this for almost 20 years now, and as part of that effort, we've curated over 20 million findings across the world's literature. We generate over 5,000 new findings every day across over 35,000 publications. We integrate over 35 both public and proprietary databases, and have actually the experience of having seen over 1 million human samples over our platform. So you can imagine the amount of variants that we've come across. And these variants are covered across over 33 different disease conditions. A little bit more about the sources of knowledge that we get it. So we get a lot of this information from both, like I said, proprietary and public sources, with the proprietary ones in pink and the public ones in white. As I said, we've been doing this for almost 2 decades now. We update our knowledge base on a weekly basis, and we have an army of MDs and PhDs who manually read over 4,000 articles a month, and we update this around 3,000 findings a day. So our QIAGEN Knowledge-base is manually curated with a team of experts to look at structured molecular data as well as disease and phenotype information, as well as clinical data, drug information, clinical trials data. We break our knowledge base into 2 sort of silos. There's hereditary disease and somatic, and then we also have our proprietary HGMD database. Now, I talk about findings, but let me just spend a little time explaining what a finding is. Imagine, if you will, that you need to take really unstructured data in the form of a scientific article and having to put that into a database. This is something that can't readily be done through machine learning because you still need to understand context, and it's very important to understand that a person who has some domain knowledge has a better way of curating this context than machine algorithms. And so that's why we employ MDs and PhDs to do our curation. Machine algorithms will probably one day be able to do this, but today, as we look at the machine learning and AI landscape, it's just not as good as expert curators. And so our expert curators take this unstructured data from literature and start to generate what is called a finding. And a finding is essentially pulling nuggets of information from a scientific article, such as what gene was mentioned in that article, what was the alteration in that gene, and some of the details of that alteration. Was there any mention of clinical effect or clinical property measured? Was there any information about drugs? And if so, what were the prior treatments and treatment details? Was it a cancer type? What type of cancer type was it? And was there a number of patients that were tested? And with the cancer type alteration, what stage of tumor was it? You take that information and you put it into a structured knowledge base. And when you have that information in a knowledge base, you can now compute across that knowledge base to find relationships that are explicit versus implied, as well as using natural language generation to generate a 1-sentence nugget of information, which we call a finding. And here's an example of a finding that is pulling these various nodes of information from the knowledge base to generate this sentence. So mutant human EGFR protein. So this is the gene information. This part, substitution, P dot, resulting in a C dot, is the alteration detail, increases the resistance, and this is that clinical effect and the clinical property measured. And then if there's drug information to AZD9291, treatment in EGFR, exon 19 activated. So this entire sentence is auto-generated because we took the time to extract this information from a scientific article into a knowledge base. And when we do that, we're essentially growing this knowledge base both from not just scientific literature, but from clinical trials, information from drug labels, professional guidelines, and various databases to accumulate knowledge into this knowledge base. And this allows not just scalability, but the ability to then take a lot of this information to validate certain criteria of the ACMG classification scheme to aid in the computation of the classification. When we talk about curating of the scientific literature, we typically talk about the clinical breadth and the clinical depth. So clinical breadth can be defined as how large is the domain of knowledge, and that can be defined as the number of unique variants or the number of unique genes that are curated. Clinical depth, on the other hand, is how deep or rich is the knowledge being extracted. So for every variant or gene, what is the total number of citations that we were able to read? And then for each paper that we read, what information are we pulling out? For example, how many clinical cases were mentioned, what drugs, what dosage, all of that information. If we take a look at the breadth of coverage, this was a survey that I did of the knowledge base back in February of 2019. And what we found was if you only look at clinically relevant variants in a database such as ClinVar, you'll find that ClinVar has around 5,300 clinically relevant genes. The knowledge base, on the other hand, has all of that, plus another 13,000 genes covered. So the knowledge base has about 3.4 times more genes than covered than ClinVar. If you take a look at the number of clinically relevant variants, again, ClinVar is just under 200,000, but the knowledge base has that amount plus another over 700,000 clinically relevant variants. So very impressive in the amount of knowledge that we have compared to just public sources. But if you take a look at a hypothetical panel of 59 genes, the ACMG 59, you'll see that the total number of variants across those 59 genes from ClinVar is around 46.5, a little more than 46,500 clinically relevant variants. If you use a proprietary database such as HGMD, there's almost twice as much, around 81,000. The knowledge base combines the ClinVar information as well as the HGMD information plus another 22,000 unique clinically relevant variants that you will find in ClinVar plus HGMD. And then in terms of the depth of coverage, how many publications per variant, you'll see that ClinVar, not all variants will have publications, so that information is not as easy to get. HGMD has around 11,500 publications for all their variants that they have across those 59 genes. But the QIAGEN Knowledge-base has almost 4 times as many as HGMD. And the reason why is because HGMD really just identifies the first mention, the first publication that mentions that clinically relevant variant, whereas the QIAGEN Knowledge-base doesn't just read that first article, but then all subsequent articles after that. So we strive to read all publications for that particular variant from the first time it's described to the present. So why is it important to have all this information? Well, in 2007, there was a lab that classified a child's SCN1A mutation as a variant of unknown significance. And because of that classification, the child's doctor didn't alter his treatment, and as a result, he died a year later. Well, the mom wanted to go back and look at those variants as part of her family planning process, and realized that back then, in 2007, there was actually 2 papers in June of 2006 and March of 2007 that could have provided sufficient evidence to classify her son's mutation in a different way. In fact, the March 2007 paper had a clinical observation of a patient with the exact mutation and the exact same disease. So she says that if that lab had read that paper, maybe they would have reclassified her son's mutation as pathogenic or likely pathogenic and possibly changed the course of treatment. And so it's really important to have access to the most comprehensive and timely knowledge for proper assessments. And what can we find, what can you improve when you have more knowledge? And we did some benchmarking, and this is an example of looking at Lynch syndrome variants. We looked at around 180 variants in ClinVar and saw and compared how those variants would have been classified using not just the publicly available information, but also QIAGEN Knowledge-base. And what we found was when you apply the QIAGEN Knowledge-base, what you essentially do is reduce the number of VUSs out of that 180 down by 27% and reclassify those VUSs to likely pathogenic, or likely benign and benign. And so more knowledge helps resolve these VUSs, which are really a challenge for labs. We've also done a similar comparison with cardiology and cardiovascular variants and found even greater reductions of VUSs by 33%. So really the value of having knowledge is to give greater accuracy in classifying these variants. Now I want to talk about carrier screening. Now, the opportunity for carrier screening currently in 2009 is very high, mainly because of higher adoption rates of carrier screening by the general population around the world, but also that there's not a lot of labs around the world that are offering expanded carrier screening. And if you take a look at genetic testing, a lot of people talk about cancer testing, but really prenatal and carrier screening testing accounts for a greater volume of genetic tests than oncology. So it's a huge market that still has a lot of growth and very few players. And in a recent conference call last year, the CEO of Myriad, at the time of the acquisition of Counsel, had suggested that we're only starting to see growth accelerating for carrier screening. And so this represents a growth of around 16% cumulative growth rate in the next couple of years. And the market has only been 25% penetrated in the US and much lower in other parts of the world, such as Europe and in Asia. So why carrier screening? Well, for the patient, for the couples, it's a low-risk test. The result of that test doesn't really affect the patient or the subject itself. All it does is provide information to inform family planning. And it has more information than NIPT. For payers, either private or government, it reduces the lifetime cost per subscriber. Last week, Novartis just got FDA approval of a new SMA drug. It's a drug that treats SMA. It's a one-time use drug, so that's good, but the cost of that one-time use is $2.1 million. So imagine a private insurance company that has to cover a newborn that has SMA. It's going to cost $2.1 million. It'd be great if they can avoid that cost by screening for parents that might harbor a risk for SMA, and then proper family planning can result in a healthy baby that doesn't have SMA and reduce that cost. For labs, it's actually a great way of accessing a high-growth market, and once you have that market, it's adjacent to a direct-to-consumer market. Funding for such a test, carrier screening, is available. There's certainly coverage by many private insurance companies here in the U.S., but we're also seeing a lot of government payers starting to pay for carrier screening, particularly in the Middle East where such rare diseases are actually pretty common. And then also, consumers and parents are willing to pay for this test out of pocket. And then finally, for labs wanting to justify the business case of getting into genetic testing, this is a great use case to get into genetic testing because of all the reasons above, but it's considered a low test. So there's a lower sort of FDA barrier to getting into this test. And then finally, with the investment that they've made in purchasing the sequencing equipment, putting together the bioinformatics pipeline, at very little incremental cost, they can introduce adjacent testing for NIPT, PGS-PGD, or newborn screening as a way to upsell the existing channels they've created through carrier screening. And then further, they can also move into oncology testing with that infrastructure that they've built out. But getting into expanded carrier testing is not without its challenges. And some of these challenges include how do you properly design a panel? And what about alternative assays to cover difficult regions? And at the beginning of this talk, my colleague Raed has talked about how we put together this panel and the chemistry used to improve the sensitivity and specificity of this panel. But as part of the design of the panel, you want to take a look at what genes and conditions are covered. And so you want to make sure that it covers some of the ACMG genes, but also the ACOG recommended conditions for carriers. You want to also make sure that whatever condition is reported has actionability. Also, because this is a business, you want to make sure that those conditions that are covered are reimbursable by both private and public payers. As well, you may want to use this panel in addition for carrier screening, maybe to possibly use this panel for PGS-PGD or even newborn screening. So the other thing that's also a challenge is before you launch a test, you need to pre-curate these genes and novel variants in a way to reduce the amount of time and effort in interpreting the variants that come out of this panel. The variant interpretation is also challenging because you need to get access to genetic expertise to interpret these variants, but also to maintain an up-to-date knowledge base so that you're always current with the literature and not run into that situation where that lab had misclassified that child's variant. And then finally, you need to do the reporting, and so to generate a report template and to generate disease statistics and reproductive risk is something that you also need to consider as part of your offering. And then finally, genetic counseling to help physicians understand the results or even to patients to understand those results. So I want to talk about the pre-curation challenge. So before launching a lab, you typically want to know beforehand all the variants that you're likely to encounter through this testing. Now, if a lab were to do this on their own, they would access publicly available sources and identify all the variants to the genes that were in that panel. And if they read all those papers and get all that information, they could probably pre-curate a lot of those genes. But after a certain period, say after 10,000 tests, they're going to start encountering more variants that was not seen in the public domain. That's one thing. The other thing is to do that pre-curation can take a long time. And if you were to look at a gene panel, a panel of around 200 genes, it could take around 7 months to do that curation. Now, if you partner with QIAGEN to do the pre-curation, we can curate much more variants, including variants that are not found in the public domain, and give you virtually the experience that you would have after seeing 100,000 variants. And even after 100,000 variants, you never get to a point of zero novel variants. And in our own internal testing, we've seen that a gene panel of around 200 genes will still generate, on average, 1 to 3 novel variants after 100,000 tests has been performed. So the advantage of the QIAGEN expanded carrier screening is we have pre-curated these 200 genes for you, so that not only do you save time, but you get a more comprehensive view of likely novel variants that you're going to encounter. So again, the reason to pre-curate is to quickly bank a knowledge of reportable variants. This knowledge then allows you to accurately classify and report these variants, and also to scale your operations. Public sources are not as complete, and that information is not necessarily of high quality. It lacks the depth of knowledge in terms of clinical case counts or functional studies. There's possible articles that are missed, and really, it takes a long time to read through all those articles and to go through all those ClinVar cases. So the panel has been matched by our expert curators, and so that panel of 200 conditions already comes with the pre-curation. Some of those genes are what we call certified coverage or bibliographic coverage. Genes that are certified coverage have the findings displayed in the user interface of our interpretation tool, which I'll explain later. And the bibliographic coverage means that we know of papers that have mention of those particular mutations. We just haven't entered that information into the knowledge base, but is that we would recommend follow-up reading. And then it's our job to maintain the currency of the literature coverage for these expanded carrier screening genes and to perform weekly updates of this knowledge. And so the value we have with this particular ECS panel is that you can let us do the reading for you, and the knowledge leverages all 28 ACMG criteria to accurately assess the classification of the detected variant. Because we've found all this information and put it into a database, we can quickly pull up the evidence and the cited article to show you the evidence and make this evidence available at your fingertips. And this results in greater efficiency and accuracy of your variant interpretation. So the other thing, as part of the effort that we can provide, is if you don't use our panel, you can create your own custom panel, and we do provide a service that will help pre-curate your panel for you. And that process is essentially identifying a list of genes in that panel. We will work with you to see what genes we already cover and ones that we don't, and quickly prepare a statement of work to curate the gap in coverage, and then finally deliver that content for your entire test panel, as well as all reported clinical cases, which is very important for triggering the BS4 and the PS4 criteria of the ACMG classification scheme. And so the value for that is by using us to pre-curate, we can reduce the number of VUS variants in your own knowledge base, reduce the future manual lookup of such variants, reduce the interpretation time, as well as increase the accuracy of variant classifications. If now you have you want to launch a service, you've launched your service and business is growing quickly, but now you're running into a bottleneck where you don't have enough variant scientists to score your variants. Well, we also provide service to help you score your variants. So as your volume increases, the number of variants that your team that needs to interpret will grow exponentially. And so it's a challenge to find scientific and clinical expertise to meet that growth. We can help with that. We offer variant scientists that can help with your variant scoring capabilities. We do this by identifying the test that you want to do, and then put together a statement of work about what established volumes and duration you want us to help you guys augment your scientific staff. We will then train our staff based on your own protocol and your own reporting policy, and then the team will then build a variant scoring team that will support your clinical activities, and then deliver a final report for you guys, for the lab director to sign off. And so the value here is to quickly scale your variant scientific team that faithfully reproduces your lab's reporting policy. It's essentially variant scientists on demand and provides you immediate access to a highly skilled scientific expertise. So next I want to talk about a case study that has benefited from access to not just the knowledge base, but the pre-curation of their variants. So Council presented this information at ASHG in 2007, where they found that their business was scaling up incredibly, and they just couldn't hire enough scientists, and so they wanted to find an automated solution that would allow their existing team to scale their operation. But before making that decision, they wanted to make sure that whatever system they moved into had the same level of accuracy as their own manual system. And so what they found was, comparing their manual effort with the QIAGEN methods, that there was a lot of effort in terms of number of articles for the variants that they came across. There was a lot of overlap between what QIAGEN and Council's manual effort found, but QIAGEN found many more articles that their team was missing out on, and then only a few where QIAGEN was missing out on. But more importantly, a lot of this information provided a classification that was almost identical to their own manual effort, which gave them greater confidence that switching over to QIAGEN will not decrease the quality of their work. More importantly, they found that the median variant search was reduced about 80%. So not only was the quality just as good, but each user spent 80% less time. Or the other way around, each scientist can scale fivefold their existing capacity. And then finally, we also provide a report for the carrier screening test. This report includes a clear result of what the final output, the test result is, a description of the test, it calculates the reproductive risk based on the result of that test, and allows a section for the lab director to sign the test out. And this is the summary page. If you go into the variant details page, it again reiterates the condition, the mutation detected, and what the interpretation of that mutation is. For diseases that are more obscure, there's a disease definition as well as the disease prevalence. It also goes into the sequencing method and the test limits, and this is interpretive material that is based on the ChiSeek expanded carrier screening panel. So if you use the expanded carrier screening panel from QIAGEN, this information is already there. You don't need to recreate that information. So this entire informatics solution really sort of dovetails with the expanded carrier QIAGEN panel to provide comprehensive knowledge, pre-curation, accurate interpretation of variants, as well as reporting. And so with that, I close out and open it up to any questions.